Hello, everyone. Good morning or good afternoon. I'm very pleased to be with you today. And I want to thank you, Francesca, for giving me the opportunity to share with you this webinar on yeast fractions differentiation with a focus on yeast cell wall. In terms of content, after a short introduction, I will articulate my presentation in two parts. The first one describing yeast in terms of variability, applications, composition, and process. And then the second part focusing on yeast cell wall as an interesting solution to sustain health and well-being of companion animals. Animals have been fed various forms of yeast and yeast derivatives for more than 100 years. Their use has recently been boosted by the growing demand for natural alternatives to treatments to provide animal health and growth performance benefits. In parallel, feed ingredients proceed from yeast fermentation processes such as distilled grains with solubles, called DDGS, has increased dramatically in recent years. Therefore, there are many types of feed additives and feed ingredients that contain yeast in various forms, but nutritionists are often uncertain about the unique differences among these products and their potential roles in animal nutrition and health. Regarding this context, where it can be sometimes difficult to differentiate feed additives and feed ingredients that contains yeast in various forms, the objective of this webinar is to share and explain information about the different types of pure yeast products available on the market, and then the yeast cell wall products as an interesting solution for companion animals. And we will see what are their characteristics and origin, how to differentiate them, and what contribution yeast cell wall can bring to animals. Let's start with yeast and especially seeing what we are talking about. Yeast are single cell eukaryotic microorganisms classified in the fungi kingdom. These microscopic fungi are about 5 to 10 micrometers in size, have a nuclear membrane and cell walls. Yeast are found in abundant quantities and are almost everywhere in the environment. Genera are known to be pathogenic, such as Candida cryptococcus, Torulopsis, and Trichosporum, while some species, such as Saccharomyces cerevisiae, Cluveromyces marxianus, or Candida utilis, provide beneficial effects. Therefore, not all yeast are equal, and only a few of these species are used commercially. There are around 60 different genera of yeasts that bring to 500 different species, and at the end, thousands of different strains, each with a unique genetic makeup in terms of morphology, yeast physiology, metabolism, activities, etc., that will bring at the end different properties and activity of the end product as fed to the animal. And it is really important to be able to make difference between product in order to know what we are using and what we can expect from those products. In terms of commercial application for yeast, thanks to its exceptional fermentative capacities, that means the capability of the yeast to convert sugars, either glucose or sucrose, to ethanol and carbon dioxide, and thanks to its nutritional properties, Saccharomyces cerevisiae is the main yeast used in food, beverage, baking, as well as in animal nutrition. And nowadays, Saccharomyces cerevisiae is used for the animal production of 60 million tons of beer, 30 million tons of wine, 600 tons of baker's yeast, and 20 billion liters of bioethanols. that leads to a huge biomass of Saccharomyces cerevisiae that can then be used also as a natural feed materials or feed additives, depending on the product we are talking about, for animals to help improve health and well-being. In terms of composition, the yeast is composed in two parts. The first one, which is the yeast cell wall, it is the insoluble part of the yeast that includes glucans, glycoproteins, mannans, and chitins. And the second part is the intracellular content, which is a soluble fraction of the yeast that include proteins, peptides, amino acids, monosodium glutamate, nucleic acids, enzymes, etc. So both fractions are different in terms of composition and also in terms of behavior. And then the benefits for the animal will also be specific depending on the fraction we are talking about. I would like to give you a quick overview of the main types of pure yeast products on the market, starting with the wool yeast cell products. And you can see that in this category, the first one will be the live yeast. It is a wool yeast that we can get after the fermentation step. Additionally, in the process, there can be an inactivation step that will bring inactivated yeast with the same composition as a live yeast, but no fermentation capability anymore. And depending on the process, the fermentation process, an autolysis or an hydrolysis step can be run that will bring to autolyzed yeast or hydrolyzed yeast. This topic will be covered in a second webinar. And during the fermentation step, yeast can be enriched in a mineral, for instance, selenium, in order to get an inactivated enriched yeast. On the other part, the second category of products is based on yeast fractions. It can be either the yeast cell wall, that we will cover today, or the yeast extract, so the liquid part, the internal part of the yeast. And it can also cover the specific association of yeast fraction by combining together different fraction, either cell wall with inactivated yeast and or with yeast extract together. That will also bring specific benefits to the animal. The production process will be specific depending on the category of product we are talking about. Let's start with the yeast cream harvesting here when the fermentation is finished and we can get access to the yeast cream. Based on that, we can get the live yeast. It can be dried at different level in order to get either dry or different level of humidity for the live yeast. If we go to an inactivation step, most of the time, thanks to a heat treatment, we will get a whole cell inactivated yeast. On the other part, this yeast cream can be lysed either through autolysis or through hydrolysis. The first one will be done thanks to the endogenous enzyme naturally present into the yeast, and the second is done thanks to exogenous and selected enzymes that are added during the lysis step. A heat inactivation step will stop the activity of the enzyme, either endogenous or exogenous, and we will get autolysis yeast or the hydrolyzed yeast. After a separation and a centrifugation step, we can get from one side the non-soluble fraction, which is the yeast cell wall, or the soluble fraction, which is a yeast extract. And let's focus now on the yeast cell wall fraction, which is a topic of interest today for companion animals. Yeast cell walls are therefore the insoluble fraction of autolyzed or hydrolyzed yeast obtained after the separation from the cytoplasmic content. And it represents around 30 to 40% of the dry weight of the yeast cell. The external layer is rich in monooligosaccharide that we can call MOS, that are responsible for insoluble bacteria binding, parietal exchanges, wall synthesis, porosity, permeability, and more widely, the protection of the yeast from the environment. It represents 20 to 30% of the cell wall, depending on the processing condition. And we can see that as it is the first line of protection of the yeast from the environment, the condition of fermentation will be key in order to grant you a good quality external layer, a good quality monooligosaccharide. The internal layer is rich in beta-1,3 glucans and beta-1,6 glucans that confers rigidity and flexibility to the cell wall, and it also contains around 1.5 to 2% chitin, which play a major role in the integrity of the cell wall, affecting the cohesion of the wall. We can also find some lipids and some monoproteins that are used especially by the mannans to fix in the cell wall. So the structure and composition of the yeast cell wall have a direct influence on their biological properties and potential activity. Each yeast cell wall product has its own specificities that influence the functionality and application in animal nutrition. To highlight the difference in terms of solubility between the yeast cell wall and the yeast extract, I would like to show you a short video, which is quite easy, that shows a test which is quite easily to do, and that assess the product behavior in water. I will start the video and comment at the same time. We will compare the solubility of a yeast cell wall and a yeast extract, putting two samples of product, either yeast cell wall on the left or yeast extract on the right, in water. The test was done with 30 milliliters of water in each tube, and we mix it with 1.5 gram of either yeast cell wall on the left or yeast extract on the right part. We mix it and we just wait around half an hour. The test is done in acceleration here, but the yeast cell wall, which is rich in monooligosaccarides and beta-glucan, is the insoluble part of the yeast and will settle through time. Whereas the yeast extract, which is rich in protein and it's naturally the soluble part of the yeast, will stay soluble all along the test. So after half an hour, we can see that the yeast cell wall fraction will settle at the bottom of the tubes, as visible in the video, whereas the yeast extract stays soluble. Just a short video to highlight the different behavior of products depending on their composition, and it will be quite interesting comparing also later on different yeast cell walls depending on their quality. Yeast cell walls can be from various origins, and it's important to know that, as it will also explain differences in terms of quality, in terms of composition and benefits for the animal. They are either issued from primary or from secondary fermentation, which also influence their properties and mode of action. From primary fermentation, this is when the yeast is specifically produced and used as food or feed ingredients, and it concerns qualitative and consistent products which are mainly used for premium segment of the market. On the other hand, secondary fermentation yeast cell wall concern yeast that are produced yeast products that are a co-product of another production process, such as bioethanol production, beer production, etc. And these secondary fermentation products are known as cost competitive, but with a high variability in terms of composition, quality, and efficiency from a product to another, but also for a single product from batch to batch. The strain and the production process that is applied will influence the purity and composition of the cell wall, and at the end with a non-negligible impact on the benefits of the yeast cell wall for the animal. Meaning that secondary fermentation yeast cell wall product must be standardized in order to control their quality and to get consistent benefits for the animals. I would like to describe a bit the process from yeast cell wall that are coming from primary fermentation. In that case, the entire process is optimized and well controlled, from the yeast strain at the beginning to the routine quality control that is applied to the end product. It's yeast cell walls that are coming from a selected production site with a unique yeast strain used during the process, with a specific and controlled production process all along the process. There is a controlled purification process of yeast cell wall and a specific enzymatic treatment applied in order to increase the yeast hydrolysis and the separation yield from the yeast cell wall to the yeast extract at the end, and to concentrate the final product in yeast cell wall. There is a quality control analysis done on each batch in order to guarantee the product composition from one batch to another one. So here we are talking about premium yeast cell wall products that are specific, consistent, and reliable. Yeast cell wall from secondary fermentation. Here, in that case, the process the yeast downstream process is most of the time not optimized and well controlled. The biomass used, the yeast biomass, is mainly byproducts from various origin that can be variable in terms of quality, in terms of origin. Most of the time, the yeast biomass is coming from different plants with various processes, and there is at the end a low level of the downstream process, meaning that yeast biomass that is that we get is that can be obtained here can be cracked more or less and then dried. So at the end, it will get access to different product quality in terms of color, in terms of solubility. We will see it right after, and a strict selection and process plus product standardization are needed in order to obtain consistent product with consistent quality when it comes to secondary fermentation yeast cell wall product, in order to get at the end consistent benefits for the animals. I would like to show you again the difference in terms of yeast strain, yeast cell wall process, and at the end yeast cell wall composition, comparing products yeast cell wall from primary fermentation or secondary fermentation, running again a quick test on the same model as the one I previously introduced, comparing yeast cell wall and yeast extract. But here we will run the test comparing three different quality of yeast cell wall: a premium product on the left, a standard product in the middle, and a low quality yeast cell wall on the right part of the screen. So again, 3 tubes of 30 milliliters of water, where we will mix 1.5 gram of yeast cell wall, depending on their quality. On the left, primary; in the middle, standard quality cell wall; and on the right, a byproduct with a low quality level. You can already see that in terms of color, there is differences between the different products. Premium one on the left and the byproduct that is darker on the right. Again, we wait around half an hour just after mixing to let the product settle in the bottom, and we will see that depending on their quality, the settling will be different in the bottom of the tube, higher in the premium product and much lower in the low quality product, meaning that the separation between the yeast cell wall and the yeast extract is more or less well done depending on the product quality. And this is really important because it's linked to the composition of the product in terms of mononucleosaccharides and beta-glucan at the end. And knowing that mononucleosaccharide and beta-glucans are the component of interest in yeast cell wall products, their content must really be analyzed in order to ensure and guarantee a high and consistent levels in both components. And those two compounds can be analyzed chemically thanks to HPLC analysis. And it's really important to see that depending on the product we analyze, we will see differences in terms of results, either for MOS or beta-glucans. You can see here on the graph two things. The first one is that depending on the yeast cell wall products that are analyzed and regarding their MOS content, we can see that there is a huge variability from one product to another one that can be collected from the field in terms of mononucleosaccharide content. And the second thing, which is visible, is an irregular content for a single product from one batch to another. Here, the standard deviation is much higher than another one, for instance, meaning that even for a single product, from one batch to another one, we have a huge variability. And the same profile of result can be obtained for beta-glucan and for protein also, highlighting how important it is to analyze the cell wall in order to differentiate products on quality criteria. And this is important, and especially regarding the mode of action and the benefits of yeast cell walls. Those benefits will be directly linked to the quality and the quantity of the mononucleosaccharide and the beta-glucans. Yeast cell wall products have three main modes of action that strongly depend on the production process and the strain background. First, thanks to their binding capacity, they help limit the development of indesirable bacteria within the gastrointestinal tract and ensure an optimal intestinal integrity, meaning that the MOS are able to bind the indesirable bacteria in the gut and to remove this bacteria through the feces, leading to a reduction of the pathogenic load in the gut of the animal. They can also favor the development of beneficial microorganisms within the intestinal microbiota, working as a prebiotic effect. And this effect would strongly depend on the yeast cell wall inclusion rate. They can also modulate the innate immune system of animals, which help reinforce their natural defenses, and especially thanks to the beta-glucans that are able to recognize the dectin-1 receptor at the surface of immune cells, such as macrophages, that will then induce a cascade of reactions thanks to immune mediators and leading to a more efficient innate immune system. And finally, cell wall help reduce the negative impact of mycotoxins thanks to specific interaction between certain mycotoxins, such as the rallenone, ochratoxin, or fumonisin, and the yeast cell wall polysaccharide. But again, those benefits are strongly dependent on the yeast cell wall quality and consistency, meaning that the strain, the origin, the production process can influence the efficacy, and different yeast cell wall brings variable results. So again, the quality control is crucial on a batch-to-batch routine in order to avoid inconsistent benefits to the animals. In order to highlight this variability of function, depending on the variability of composition, I would like to show you An example results from an in vitro test that highlights the variability of functions. So the yeast cell wall variability of composition reflects the variability of function, and at the end, the irregular benefits for the animals. And let's take an example in regard to immune function properties. And for that, we had a look to TNF-alpha and interleukin-10, which are two immune mediators that reflect the capability of yeast cell wall to modulate the innate immune response. These cytokines are induced by the receptor, for instance, dectin-1, present at the surface of immune cells, such as the macrophages, and they induce a cascade of reaction leading to animal immune response. So paying attention to immune mediators in vitro is a robust model to assess yeast cell wall immune properties. So with this in vitro model, we compare yeast cell wall 1 with yeast cell wall 2 and with a positive control, which is a zimosan, often used as a positive control due to its high capability to induce immune modulation. So depending on the dose of the product, zimosan or yeast cell wall 1, yeast cell wall 2, we measure the level of immune mediators that are produced, either TNF-alpha in gray or interleukin-10 in red. And we can see that depending on the product, the level of immune modulation is strongly different, where product 1 is closer to the positive control, and the yeast cell wall 2 leads to a much lower level of immune modulation. Again, all yeast cell walls are not equal, and it's really important to pay attention to the composition and quality of the different products. And there is a strong need for standardization, in order to ensure consistent benefits for the animals. There is a last category of products in the family of yeast cell wall products that I would like to quickly introduce, which is the exposed beta-glucan. It is a specific category of yeast cell wall where an additional step is applied during the yeast cell wall process, right after the centrifugation leading to yeast cell wall separation from yeast extract. And right after this step, an enzymatic or chemical treatment is applied on the yeast cell wall fraction in order to remove the mononucleoside layer. The finality of this additional step is to enrich the final fraction in beta-glucan. And then the final product obtained contains a high level of exposed beta-glucan for specific immune system modulation functions. And these enriched beta-glucans are also of interest towards certain mycotoxins, and especially towards zearalenone. So this is a bit different from the yeast cell wall product containing mononucleoside and beta-glucan. Here, the process is oriented in order to obtain a product enriched in beta-glucans, and where mononucleoside are removed. To conclude, yeast represent a flexible and valuable source of functional and nutritional ingredients for animals. And each yeast product has its own characteristics and beneficial effects on animals, depending on the composition. The hard work of characterization and yeast selection is needed in order to identify the best strain regarding their potential benefits. And a strict control of the process is key, from the strain selection at the beginning to the fermentation and the post-fermentation steps, including the drying that will lead to different composition and benefits of yeast cell wall product. So yeast cell walls are of interest, but all yeast are not equal, and the quality must be monitored and controlled in order to guarantee the quality of the cell wall product. Before finishing my presentation, I would like to introduce the next webinar where you will learn more about the hydrolyzed yeast and their nutritional functionalities. So do not forget to register to this second webinar that will be totally complementary to the one I presented you today on the yeast cell wall part. I would like to thank you very much for your attention.
Thank you very much to you, Bruno, for having so well explained the differences between yeast fractions. And now let's spend just a few minutes looking a bit deeper on how yeast fractions can be used in companion animals' nutrition. So we have seen a bit in details with Bruno the yeast fractions, the different yeast cell wall components, how do they work, and how they differ in composition, mode of action, and thus benefits that can bring to the animals. But what I would like now to do with you is to show how Lallemand solution fits in each of the mentioned categories. And we will, as Bruno did, focus on yeast cell wall, but a bit later I want to spend a few minutes with you also on the specific association of yeast fractions. So within the category of yeast cell wall, Lallemand can offer three products: Agrimos, Optiwall, and M-glucan. Let's see how these three products fit in each of the different categories. Yeast cell walls can be, as we have seen, from primary fermentation, which means they have been produced through a optimized and well-controlled production process. So there is a selected production site, there is a unique yeast strain, there is a specific and controlled production process, which end up, after the quality control, into a primary yeast cell wall. And that's the case of Agrimos, a premium yeast cell wall product, which means that since from the first step, so from the yeast strain selection, it has been produced specifically for human and animal nutrition. Then there are the yeast cell wall from secondary fermentation, which are yeast cell walls coming from the byproducts of brewery and distillery. In this case, products can come from various origin, from different biomass, so there is a lower level of process control. Nevertheless, it's possible to have a strict selection of the origin. It's possible to have a process and product standardization in order to ensure a consistent quality product. That's the case of Optiwall. What Optiwall offers is an yeast cell wall from secondary fermentation, but in which the production process is standardized, well controlled, and so ensuring a consistent quality of the product. The last category of yeast cell wall are the yeast cell wall product that has exposed beta-glucan. So it's an additional step that can be applied during the yeast cell wall process in order to remove the mannanoligosaccharide layer and make a product that is rich in beta-glucans. So it's what is usually called purified beta-glucans. Beta-glucans can come from different origin. They can have bacterial, fungal, yeast, or cereal origin. But if you can see, the structure is quite different. And you see, for instance, the bacterial one or also the cereal one are a linear structure, while the fungal has this branch like beta 1.6, beta 1.3 glucans, but they are short, while the yeast is the only one having a long beta 1,6 and beta 1,3 glucan. So this different structure ensure much better efficiency of the product. Lallemand can offer a purified beta-glucans product called M-glucan. So we have seen that Lallemand can offer primary cell wall products, Agrimos, secondary cell wall products, optimal, and also purified beta-glucans, M-glucan. In case you would be interested in getting more information about these 3 products, don't hesitate and contact us. Now I would like, as I promised, to spend few minutes in something that is a bit more innovative. Not a bit, I would say a new generation of yeast derivatives, because the yeast cell wall products that we have seen, they are already on the market. There is already a good offer of this product on the market, but we have seen that there are different qualities, and it's important to be able to select the right one and to be sure of the quality and consistency of the product. And Bruno has been very good in clearly explaining and helping us, giving some guidance on how to choose the right yeast, because as said, not all the yeasts are the same. But what about the specific association of yeast fractions? That's true. It's possible to take different yeast fractions, combine them together to have a specific and well-targeted benefit. That's the case of Young Yeast Association New Generation. Young is a new generation of yeast derivatives. It comes from an R&D project that has developed an innovative product combining inactivated yeast fractions from 3 different strains. Each of these strains is produced through a dedicated process, and those 3 strains has been selected and combined in order to provide superior binding capacity and a synergistic, broad and balanced immunomodulation. We can see now few information and a few trials that has been run in order to support what I just said, the benefit that I just mentioned. Let's start with the superior binding capacity. In an in vitro trial, we have been able to compare the binding capacity of Young versus the binding capacity of a good quality moss product. And as you can see, we have used the commercial recommended dosage of Young, which is 800 grams per ton of pet food versus the 2 kilo moss that is usually used. We used to assess this comparison, we used strains of Escherichia coli that were isolated from dog and cat feces at different life stage, puppy, kitten, adult, senior. And it's important to remind that all the 5 strains were collected during enteritis, and those coli were identified as the causative agent of diarrhea. From the result, you can see that Young were able to agglutinate plus 19% of Escherichia coli, even though Young inclusion was 60% lower than moss. But nevertheless, it led to a significantly stronger reduction of coli if compared to a good quality moss. So we were really able to show the superior binding capacity of Young versus a good quality moss. In addition to that, we performed trials in parallel, in vivo and in vitro trial at the University of Bologna in Italy. For the in vivo phase, we used 16 healthy adult dogs in a trial that lasted 28 days, and in which we had 2 groups, the control group and the treated group that was supplemented with a pet food containing 800 grams per ton of Young. We look into the IgA, fecal IgA, and we have seen that in the Young group, the IgA level was 59% higher 21 days after the start of the supplementation and 56% higher after 4 weeks, so 28 days. So it was really confirming the great immunomodulatory ability of Young. In terms of fecal pH, we have seen a drop after 21 days and also a trend at 28 days that are reflecting the reduction of pH at colonical luminal level. And so it's such an acidic environment, it's really helpful because it inhibits the development of undesirable bacteria and helps stimulating digestion, which is really beneficial for the host gut health. On the other side, in the in vitro phase of the same trial, we looked into the microbial population, and again also in the pH, because we wanted a confirmation of what we have seen in vivo. And so it has been used a methodology published by Biaggi and Pina in 2016, in which there were the usage of 5 flasks in an aerobic chamber containing canine fecal inoculum plus undigested pre-digested extruded diet. As I said, we look at the pH and we got a confirmation of the reduction of the pH that confirms what we have just seen in vivo, and also the positive effect of yang supplementation on gut health. But what is quite interesting is also the microbial population. So in the control, in the treated group, in the yang supplemented group, we have seen an increase in the total microbial population. And looking deeper into the bifido and lactobacillus, so the beneficial bacteria, we have seen an increase of 10 and 14% after 24 hours, and similar value also for the lactobacillus in the yang supplemented group. So it was showing really the beneficial stimulation of yang on the microbial population. So with this in vitro trial, we were able to show the digestive support that yang is able to offer together with immunomodulation and superior binding capacity. And so to conclude, yang is a new generation of yeast derivatives, is a synergistic alliance of specifically selected inactivated yeast strains that helps to reinforce the immune system and to support gut health. So thank you very much for your attention. And now let's go back to Meet the Expert webinar, because it's time for the question and answer session.